All right, so welcome everyone. Thank you so much for joining us today. So today is week 2 of our 4-part series in partnership with OMPRN, the Ontario Molecular Pathology Research Network. So we're so excited for part 2. We had lots of great feedback on last week's session. So if you're tuning in for week 2, we're glad you're here. And there's lots of exciting stuff to learn today. So I will pass it over to Kester, one of the leads at OMPRN, to introduce our speaker for today.
Thank you so much, Vanitha. Good afternoon, everyone. I am pleased to introduce today's speaker, Dr. Rola Salib. Dr. Salib is the genitourinary pathology site lead at St. Michael's Hospital, which is part of Unity Health Toronto. She is also a clinician scientist at the Ontario Institute for Cancer Research and the Keenan Research Centre, as well as an assistant professor at the University of Toronto. Dr. Salib runs a research lab where her focus is on genitourinary pathology and kidney cancer. She and her team study renal cancer and are working hard to find ways to make molecular tests for cancer more accessible. In addition to all of these amazing things, Dr. Salib is also a longstanding member of the OMPRN community. She was the recipient of several research grants and is a regular presenter at OMPRN conferences and events. And for all of those reasons, I am thrilled to have her here with us today to help explain the basics of molecular pathology and why it matters for patients and their families. So please join me in welcoming Dr. Rola Salib.
Thank you so much, Kester. So I'm going to stop my share. Rola, feel free to pull up your slides.
Thank you, Vanitha and Kester. Thank you for the lovely introduction. I'm not able to share my slides.
Just give me 1 second. I think that's on me. There you go.
Yeah. Okay, thanks. Yeah, I think this is okay.
Perfect.
Good. You see my slides?
Yep, and we just need to go full screen. Perfect. Thanks, Rola.
Okay, thank you. Thank you very much. It's really a privilege to give this talk. So I'll be talking about what is molecular pathology, a brief introduction to what is pathology in cancer in general. So just what is pathology first? So pathology is the examination of tumors under the microscope. We receive tissues in the lab, either through a resection or a biopsy. And if it's a biopsy, we usually examine the entire tissue. If it's a big resection, as in a surgery, we select areas and we examine these under the microscope. And this is how they look under the microscope. So eventually it's processed in the lab to have tissues on slides as such. And these are stained with our traditional blue and pink stain. And this is usually how it looks like. And this is how we see tissues under the microscope. And our job is to examine that and give you our reports. And what do we report on? We report on whether it's cancer or not. If it's cancer, or if it's not cancer, what is it? And if it's cancer, what type of cancer it is? Because there are many types of cancers. Each organ has its own variety of cancers that arise in it, and that matters, as I would explain more. What grade is it? There are grading systems for every single cancer that exists, and they're all different. Is it like high grade, low grade, which means is it like a biologically more aggressive cancer or not? The extent of cancer, what stage is it, which means, for instance, depending again on the organ and the site, is it like confined to the organ or is it outside of the organ, which reflects into the final staging of the cancer? Whether it was fully resected by the surgery, if it's a surgery and not a biopsy, whether it was fully resected as in margins were negative or not, whether they're lymph nodes and biomarkers as well. We do report on certain biomarkers for certain cancers. So this is a generic surgery pathology reports from the internet, which shows like sections. I just wanted to show basically how it looks like, but basically what we comment on is the diagnosis. We have the final diagnosis line. we have a comment sometimes that explains a bit more what stains or how or what this cancer is. If it's a funny cancer, for instance, or not a rare cancer, sometimes we communicate in a comment to the surgeons or to the oncologists what we're seeing and why we're seeing it, or what the implications of this cancer is. Microscopic is not always a section we fill because it's a detailed section that only pathologists would understand. And the gross description is how it came and what sections we took. And very importantly, also the synoptic report, which has a lot of details that the clinical team then uses to complete treatment, management, next steps, and all that. So this is what pathology is. Let's talk about the molecular, the genetics of cancers. Just I want to start by saying or clarifying that there is a phenotype and there is a genotype. If I give you two sets of DNAs and I say there is more than 98% similarity between the two sets of DNAs, you think they're very, very similar. But then I proceed to tell you one DNA set comes from a monkey and the other DNA set comes from a human. And this is what I mean by phenotype and genotype. Phenotype is how it manifests, like the look, the final look that this DNA provides. So in one instance, this is the context is a monkey, and the other instance is the context is a human. And unless you know the phenotype, which is how the final look is, it's kind of meaningless to study the DNA. And this is exactly how it is with molecular pathology. We start with a microscope because the microscope is how the cancer looks under the microscope. This is your phenotype. And unless you know how it looks under the microscope, the main type of cancer, the main subtype of cancer, it's kind of not useful, or it can be even harmful to go into studying the genetics of this cancer without understanding the final look of this cancer. So in context, it's very important always to study the genotype in the context of the phenotype. How does it look like under the microscope? And this is very important, I would say, because you can have the same exact gene mutation that in one cancer, it would imply that this cancer is more indolent or well-behaving, and in another cancer, it would imply that this cancer is more aggressive. So it's very important to take it in context of the phenotype. What is the cancer under the microscope? And that's why we start by studying the microscope. What is the cancer first? Now, what is molecular pathology? Molecular pathology, as science has advanced and we started to study cancers more on the molecular level, we started to understand more. And this is really what molecular pathology is. It's a study of the cancer on the level of the molecules. And there are really three main molecules that we do study: the DNA, the RNA, and the protein. Mostly the DNA and proteins, I would say, but we also do study RNA in some situations. And this is the sequence of the significance of these molecules. So you have your DNA, and the DNA produces copies of RNA, and the RNA produces your final ultimate protein. So you can study it on the final level, the protein. What is the protein that is expressed, or like what's the protein that's present in your cancer? Or you can study the RNA profile, like what RNAs are there in this cancer, or you can study the DNA itself and what genes are perhaps abnormal in your cancer. So it's multiple levels. So molecular pathology is not one level, and it's quite complex actually. So let's start with the proteins. The proteins is the final ultimate outcome of your molecules, if you may. So for proteins, for a very long time now, we've been using something called immunohistochemistry. And we do incorporate immunohistochemistry, or immunostains as we call them, in the pathology report. Sometimes we just call them IHC stains. They're part of the pathology report for at least 2 decades now, if not more. A big part of what we do, and we don't call it really molecular pathology, even though technically it is part of molecular pathology, because we do examine certain molecules by using immunohistochemistry. The way immunohistochemistry works is it looks for a specific protein. So each stain looks for a protein. If the protein is present, it gives you a brown color. If it's not present, you don't have a brown color. Like, so for instance, these cells here are blue, so they're not expressing this protein. But these cells, which are the cancer cells, have that protein that we were looking for. And this protein can be expressed in different areas. Like, for instance, these cells, the membrane of the cells are brown. This is what's expected for this protein, versus these cells on the right, the nucleus is brown. So the protein is present in the nucleus. Without going into details, just suffice it to say that it's a big part of what we do. It helps us characterize cancers, sometimes helps us characterize the aggressiveness of a cancer, and they're very important as part of our pathology work. We can have cases that don't require any immunohistochemistry, and with the complexity and as our understanding is increasing about cancers, we find ourselves actually using immunostains more and more. For instance, I do a lot of kidney cancer, so I'm going to use kidney cancer a lot as an example. For instance, right now, kidney cancer is 22 subtypes. The main category, and not even all categories, the main category of kidney cancer is 22 different subtypes. So I find myself sometimes doing panels of stains up to 10 and 15 stains, and even more if it's a very complicated cancer that we're trying to understand what it is. So this is part of what we do. It's part of the pathology report. It is a molecular test. It's just we don't call it that, but it is a molecular test on one level. And then now we have the ability. So immunostains were there for quite a while. Now we're starting to do more. What we're now really calling the molecular report, or incorporating as part of our molecular report. Let's talk about the DNA itself, because that's the second most common molecular marker that we do test. So you have your cells. Each cell has your 46 chromosomes, and then each chromosome, you can spread it out into DNA. Each chromosome is formed of the DNA. And the DNA is formed of genes, many genes. And each gene usually gives you, as I implicated before, RNA and then protein. So now we often study the DNA. Again, depends on the context. Sometimes we need to count the chromosomes. It matters in certain cancers, like certain blood cancers, for instance, or brain cancers. It matters if you have a whole chromosome loss or gain. On the left, this is a bit of an old method, counting, which we usually don't use. We usually use more advanced techniques, as the one on the right, where, for instance, this is a chromosome here, and then suddenly you have an area in that chromosome that drops. So we have a loss in part of the chromosome. And it matters. Again, depends on the context. Not every cancer, it doesn't matter in every cancer, but there are certain specific cancers where it would change the classification, change the treatment, change the outcome, expected outcome, and that's why we test it in certain cancers. That's one level where we test the DNA. We also test the genes, and this is really the more common molecular test that we're doing nowadays. Many hospitals or the molecular centers, I would say, that have a genetics like center department, usually they have like a panel of genes that they would test in cancers. And now we're looking at the DNA at a very zoomed in level, very, very high resolution. So we're looking at the level of the nucleotide. So genes are formed of nucleotides, hundreds to thousands. This is the smallest molecule now in the DNA. So we're looking at the level of each nucleotide by a technology called next generation sequencing. And we're looking whether there are these nucleotides are changed, substituted, removed. Very high resolution tests. Sequencing is something that's now becoming more and more common. This usually tests a panel of genes. And it has implications sometimes, and I will explain more. And then there's another level of testing for the DNA, which is called the methylation. Now we're not looking at the DNA itself. We're looking at certain chemicals or tags that tag on the DNA. And if they tag in certain spots, this methylation can affect the gene job or the gene function. So some of our tests, again, very in limited cancers, this is important. Like for instance, in brain cancer, this DNA methylation can affect the response of patients to certain treatments. So we do test it in certain cancers as well. And then lastly, maybe the least common, but we still do test, are the RNAs. There are, again, multiple levels of testing the RNA. So one of them is fusions. Certain cancers are have what they call fusion events, and we do test for that with RNA, and it can help us determine what type of cancer it is in certain type of cancers. And then there is the RNA gene signatures. This is more available commercially usually. The most famous gene signature test is really the Oncotype DX. That's the one used more clinically. There are, for instance, similar gene signatures for prostate cancer, but not as commonly used or clinically. But what it is it tests a number of RNAs signatures in the cancer, and what it tells you is it would tell you, for instance, whether a tumor can likely recur or not, the likelihood of recurrence, or would this cancer benefit from chemotherapy, for instance, or not. So it can also help guide treatment decisions. So I didn't really mean to complicate matters. I just wanted to show how big molecular is. So when someone says, let's test this cancer molecularly, let's perform molecular testing, molecular is really very big, and we almost never test everything, never really. What we're doing really more and more commonly now is to do some sort of a targeted molecular testing, is to look at certain areas or certain things on the molecular level to test that can give us an answer that would help next steps for the treatment of patients, for instance. But we're rarely really ever testing everything. And what it helps with, as I had implied, is sometimes it helps with the diagnosis. It helps us know what it is. Some cancers we cannot currently diagnose without doing this molecular testing event. Sometimes it tells us how a cancer would behave. It can predict outcome, whether it's a more aggressive or less aggressive cancer. And sometimes it helps guide treatment decisions. It can tell you, for instance, whether a cancer can respond to a certain therapy or not. And that's really why we do molecular. And as I said, in the context of what is the cancer under the microscope, because molecular testing without knowing the phenotype is not very useful. So I would give an example of a case. It's a scenario that happens. It's not an actual scenario, but not an actual patient, but an actual scenario that happens commonly in kidney cancer, for instance. So our patient's scenario is a 27 female. She started to develop back pain and then had CT imaging, and they found that she had a 4-centimeter kidney mass. And then that mass was biopsied, and we received the biopsy in pathology. And it looked as such under the microscope. And this brought up the differential of a number of, as I said, there are 22 entities in kidney cancer. So this brought up a big differential, like we had to work it up for whether it's A, B, C, D, for instance, like 4 main, depending on that look. We had to, like, there were 4 main diagnoses that we thought about, and then we worked it up more with immunostains to determine exactly what type of cancer it is. And we suspected a certain cancer that is called FH deficient renal cancer. And this entity specifically is what we call a molecular defined entity. Like, we cannot really confirm the diagnosis unless we do molecular testing to confirm that this gene is actually mutated in the patient. What's the significance? The significance is this is a very aggressive type of cancer. This was a biopsy. This is one significance. So first of all, it would determine next steps for the patients. In kidney cancer, if it's a small lesion, it was a 4-centimeter lesion. They could sometimes, if it's a well-behaving cancer that doesn't grow much, they can just watch and wait, or they can remove part of the kidney with the cancer and try to preserve the rest of the kidney. Sometimes there are new technologies where they could even just burn the cancer with like radiotherapy or freeze it and not even do any surgery. So there are options. But for aggressive cancers like this one, if this is FH deficient, the better treatment would be total removal of the entire kidney, because this is a very fast growing, very aggressive cancer. So finding the right diagnosis, and this is a molecular defined entity. So in this case, we have to confirm it with molecular test. So arriving at the right diagnosis can help the next steps in management. So this is one aspect where this is important. The other aspect, so we had to do molecular testing for this. And the other important aspect is this mutation can be hereditary. And this is very important in this scenario. So if this is hereditary, there are implications to the family. So the next step would be to test that patient themselves, test their blood, not just the cancer. If the patient has inherited this gene mutation, and perhaps other members of the family have this gene mutation. So there are implications to the family and to the patient. To the patients themselves, if the patient has this hereditary mutation, they are liable to get other cancers. So they should be watched. They should be followed thoroughly in case they develop another cancer. And for the family should also get tested. And if they find that they have that mutation, they should also be followed very carefully. And that can help. That can help because if you're actively looking for a cancer that can develop, you can detect it early and you can treat it early and you can save the patient's life. So a lot of implications in a case like that, and molecular is very important in such a case. The other aspect that can also be important is that treatment of this cancer, if the patient develops metastasis, if the cancer grows outside of the kidney, these patients get systemic treatments similar to chemotherapy or the newer forms of therapy. And not all current therapies works for this type of cancer. And there are clinical trials, for instance, specific clinical trials that a certain combination of drugs can work better with this type of cancer than another. So you have a lot of implications in a case like this. So molecular is very important in something like that, where implications to the patients, implications to the family, implications to the treatments. So it can guide treatment. So molecular can guide treatments, and as examples to guiding treatments for certain drugs is that it can indicate, for instance, that a patient can respond to a certain drug. So that would, so then it's better to give that patient that drug, or the patient can be resistant to a certain drug. And then it's better to spare them that drug because it's likely not going to have effects, and patient can develop a lot of side effects from that drug. So examples is a mutation, for instance, that causes a cancer to be resistant to a specific treatment. Or for instance, in the case of immunotherapy, currently immunotherapy is one of the modalities that are very helpful in current age in fighting cancer, sometimes expressing a certain protein. And we sometimes look for these proteins just by immunohistochemistry just expressing certain proteins can indicate that a patient can maybe respond better to immunotherapy. Or sometimes you have a protein that the protein itself can be blocked by therapy. You have a drug that actually blocks the actual protein. So who tests molecular? So not all hospitals have the ability or to do molecular testing. So immunohistochemistry is pretty much in every pathology department. Some pathology departments are bigger than others and perhaps have a bigger immunohistochemistry menu, but it's accessible. Immunohistochemistry is really everywhere. Molecular in Canada, for instance, is not everywhere, usually big centers. So if a patient has a cancer that requires molecular testing, that cancer would be referred to be tested to one of the big centers. And Cancer Care Ontario luckily covers many of the markers that we need. Not all of them, but many of them, many of the significant markers. So hospitals are able to do this and get coverage. This is a sample, for instance, from a molecular report for prostate cancer. So prostate cancer that is very aggressive, high-grade aggressive, we have certain criteria that if it's a bad cancer, we would do molecular testing. And the significance is if it has a mutation in any of these 4 genes, for instance, a significant mutation in any of these 4 genes, the patient can respond to a certain drug. In this case, for instance, the patient was negative. He didn't show any mutations in the 4 genes. And what we're looking for when we look at the mutations are genes with a clinical significance or potential clinical significance, which means so not every mutation counts. Some mutations are, for instance, proven to be not useful. They don't make any difference to the treatment given. And some mutations we don't know. It's still early and we're still discovering more about them. And some mutations are known to have an effect and likely to make a difference when you give the patients the treatment. So this is what it means when we say negative for disease-associated tier 1 or 2. Tier 1 or 2 are sort of like the significant or likely significant mutations that are going to affect treatment. So this patient didn't have any of the genes mutated, so likely not to respond to this treatment. And this is important information where the patient's clinical team can look and say, okay, maybe not give him this drug, but give him another drug. Okay, lastly, I want to talk about liquid biopsy. Liquid biopsy is really an evolution in the field. I've been talking for the entire presentation about the importance of doing molecular in the context of the right pathology and after testing the actual tissue. So liquid biopsy is not testing the cancer tissue itself, it's actually testing body fluids, likely blood. So what we do is we examine, let's say, blood for material that comes from the tumor. Likely, in most situations, the most common is we're looking for fragments of the tumor's DNA that has shed from the tumor into the blood, and we're looking for these. And this can have benefits. There are implications. It's still an early field, I would say. It's not the answer to everything yet, not by any means, but we're getting there. Many applications. So this is like a diagram of how it happens. So here, like these are intact cells, and these are cancer cells that are dying, and all cancers have a blood supply. And this is how the material goes to the blood. So cancers, to survive, they have to have blood going into them, blood supply going into them. And this cancer, for instance, is dying. So dying cells can release fragments of their DNA in the blood. And these are the fragments that we look for, try to collect, and try to examine to understand more. Benefits. So the benefit is, so for instance, if you're looking, and I'm going to speak about each benefit and its caveats really. So really, this is useful if it's a type of cancer that grows fast and dies fast. Not all cancers are like that. cancers that grow fast and outgrow their blood supply, die, and then release fragments of DNA. Some cancers are slow growing and don't really shed as much DNA in the blood, but for the cancers that are fast growing, this can be useful. So for instance, you can look for tumor recurrence. If a patient had cancer, had surgery, had chemo, and you want to follow up whether and patient is cancer free, and then you want to follow up to make sure the cancer doesn't come back. Now, the traditional methods right now for following up on cancer, whether it came back or not, is really imaging, like we do a CT or MRI. So what if there is another method that maybe would pick up the cancer, the cancer has come back earlier. And again, this is not applicable to every cancer, does not always produce benefits. That's why it's not clinically used, except in very limited scenarios and not readily accessible everywhere, because we're not yet there. But the promise is, as we're getting better and better with technologies, perhaps we can pick up very small quantities of DNA in the blood, and this can help us even earlier than CT and MRI to detect the cancer that has come back. Early tumor detection, this is very useful, let's say, in families that inherit cancer genes, and they have a high likelihood of developing a number of cancers, and they are on surveillance, like surveillance by imaging, physical exam. This is a modality that can be helpful. If you can do a blood test, non-invasive blood test that can detect cancer very early, and you can know, oh, we've detected a small fragment of cancer, let's look where it is. This can be very helpful. It can help in guiding therapy if it's a patient that has cancer and is being given chemotherapy, for instance, and then the cancer develops what we call resistance, a resistant mutation. This is actually something that we apply right now for lung cancer. Lung cancer patients often get this treatment that they respond to, and then after a while they develop a certain resistance because of a gene mutation. And we do actually use liquid biopsy blood to try to find fragments of the DNA. It's a very sensitive test, and we're looking very specifically for one gene mutation. And if we find that mutation, that means that the patient has developed resistance to this cancer. So this is actually one actual clinical application that we're using right now. And of course, it's non-invasive. Well, as I said, it's early. It's very limited centers. We have and there are really two things we test for. We either can take the entire cell-free DNA, like the tumor fragments, and test them for everything, like a panel of genes or a panel of cancer genes, or we can be looking very specifically for a certain gene, as I mentioned in the lung cancer. The targeted one, where we're looking for a specific gene mutation, is currently something that we're using more in practice. The comprehensive one, where we're looking for a panel of genes, is something that is currently present in limited centers. I believe William Osler just starting to test this. But we're still in the early stages, not applicable for every cancer, and as to my knowledge, not yet covered by perhaps Cancer Care Ontario. Okay, and this is the end of my presentation, and I would be very happy to take questions.